Today I'm going to talk about the research project that we are doing at CMU. During the presentation, I'm going to show you a lot of time-lapse movies because I'm very interested in understanding the morphogenesis. And maybe you have seen fruit flies like this one. Let me label it. Possibly in the kitchen of your house during summer and you find them annoying. I want to tell you that due to the rapid growth of genetics and cell biology, now in many labs, our fruit flies look like that. They are fluorescent. And this allows us to follow the cells and track the cells during development. So for example, here you can see a movie of a wing of a fruit fly, and we can see what is happening during development. Here we have another example of the formation of an egg, and here you can see an example of early embryonic stages in fruit fly. And we are not only doing that. In developmental biology, we can also use, you see here in the bottom of the screen, you can see computer simulations. And this is exactly what we are doing at CMU. We are using time-lapse movies as well as computer simulations to start to understand the cellular process happening in the leg morphogenesis. And this is one type of project that we are following, that we are exploring at CMU. And I'm going to show you some examples later, talk more in detail about it. And we're also doing another type of projects. Here, you can see in green, both on the top, you can see the expression of GFP, a fluorescent protein, in cells that are expressing a gene called RASB12 that is involved in multiple cancers, and we are using fruit flies to study that and to study the cell dynamics of cancer. And in the bottom, you can see I'm just labeling the cells so you can study it in more detail. So it's much clearer for you to understand what is happening. And as you see, during development, those cancerous cells start fusing together. So if you see our schematic in the beginning, in the beginning, all these tumors are separated, but when we study all these cells are fusing together, and this is a pretty cool example of how cancer can be tracked using fruit flies. And I have a student that is working on other cell behaviors, other cancer cell dynamics, and I will show you a video of what he's doing. Now I have shown you this time-lapse movie multiple times. Now I want to tell you a little bit more of what we do. So this is a time-lapse movie of a fruit fly leg, and the region that we are imaging is this wide region that is very small, that is called the first dorsal segment. We are studying this row of cells in yellow that are changing in position, that are rotating. And also we are studying what happened around the sex chromosome, around this group of cells. And we want to know what are the cellular processes involved in this change of position. And we find that this is a great system to study the relationship between between physics and biology. And this is something that we are pursuing. Finally, to finish, I found that when my students are involved in these research projects, in addition to develop transferable skills, they're able to have a deeper understanding of fields like developmental biology, evolution, genetics, as well as cell biology. And I'm really in favor of allowing my students to write their own publications. And these are some examples. And we are hoping that this semester some students develop more projects to continue writing more small research publications, because I have seen that this allows them to improve their resumes and their application into grad school, medical school, or even an MBA, because it showed that they have critical thinking. Now I would like to show you one project that we are doing, and later if you have many more questions, if you have any questions, just let me know.